So let's talk about what happens between neurons. Now, remember my computer charger? It's now plugged into the wall and connected to the computer. What would happen if I took scissors and cut the charger wire? What would happen? Vincent, what do you think?
I'm not really sure.
Okay, so let me ask you more plain. If I cut this electrical charger, the charger wire, can I charge my computer with the wire anymore?
No.
No.
No, right? So the ability to pass an electrical current from the light socket all the way to my computer has been interrupted. So you might ask yourself, hey, wait a minute. You said earlier that neurons have gaps between them called synapses. So neurons operate like cut electrical wires. How is it possible that a message gets from the body to the brain or vice versa? Right? How is it possible that this communication occurs? You said, hey, inside the cell, it's electric, and you cut that electrical wire. How is that impulse moving between the cells? So that is also a cool finding. The answer is pretty simple. Remember in the terminal button, I mentioned that we had these chemical messengers, which we refer to as neurotransmitters. Well, these chemical messengers get released into the gap, get released into the synapse that is the space between neurons. And what does it do? It binds to a receptor site on the other side. And then that chemical messenger alters the property of the next cell and causes that input in the next cell. And I'm going to explain all of this as we go. But if you're looking for a simple answer, how do we get communication between cells? It's that chemical transmission. It's our neurotransmitters at play. So when we think of two neurons, let's say my left hand is a neuron, my right hand is a neuron, and the space in between is what we call the synapse. Well, we have neuron A, which we're going to refer to as the presynaptic membrane. That's the first neuron, the original neuron. And then we have neuron B, which is our second neuron, and that has to receive a message from the first one. So the terminology we use for the first neuron is the presynaptic membrane. That is where your axon is, where your terminal button is, and that's where we release the neurotransmitter from. Now, there's neurotransmitters are in like pouches or sacs, which are called vesicles. And there's a process of releasing the neurotransmitter. So the presynaptic membrane releases the chemical. The postsynaptic membrane is on the other side of the synapse, hence why we say post. Post means after. Postsynaptic membrane after the synapse, right? Well, that's going to be the receiving neuron. Do you remember what part of the cell receives input? From the previous video, what part of the cell is responsible for
Dendrons?
Good job. Receiving input. It's the dendrites. So on the other side, on the postsynaptic membrane, we have a whole bunch of dendrites. It looks like the tree, the arbor of the tree that we talked about. And what's on these dendrites? Receptors. And like a lock and a key, each neurotransmitter fits into a receptor. And depending on whether that neurotransmitter is excitatory or whether that neurotransmitter is inhibitory, that next neuron is going to be depolarized or hyperpolarized. It's going to increase or decrease the likelihood of that next neuron firing. But that's how the messaging occurs. A neurotransmitter is released into the synapse. It binds to a receptor site on the postsynaptic membrane, and then it alters the electrical properties of the postsynaptic membrane or the second neurotransmitter. Now, going back to the analogy of an email system and a server, we have a spam folder. We have a or deleted mail folder. In the synapse, there is a process for marking a neurotransmitter for destruction, which is pretty cool, right? This is spam, delete it, right? And then it gets rid of it. So that's one process of getting neurotransmitters out of the gap. But there's a second process of what do we do with this unused chemical? We can't leave it in the synapse, because the longer it stays in the synapse, the more likely it's going to bind to the postsynaptic membrane and cause erratic firing of neurons. So the neurons that aren't marked for destruction, we have a process called endocytosis, which is just a fancy way of saying reuptake. The presynaptic neuron reabsorbs some of the unused neurotransmitter and processes it for recycling. The body is a very brilliant and efficient system, right? The fact that we have a process to protect against excessive firing of neurons by marking it for destruction or reabsorption, that's a pretty cool system. And I guarantee you, you don't think about that. Your body just does it. Your brain just does it. So just to give you the process, right? So if we were to look at a process, so an endosome is responsible for preparing your neurotransmitter, packaging it, and sending it to the terminal button. And then we send it to the base of the terminal button. And remember, I said it's in sacs or pouches. When it gets to the base of the terminal button, the pouch opens. And these black dots are the neurotransmitters in this picture. It releases it. And then on the other side, what you're not seeing is another neuron to receive that chemical. Whatever is not used, you'll start to see there's a process of reabsorption. So it swallows it back up, brings it back along microtubules, and then it prepares it for recycling, right? So that's the process of the release of neurotransmitter and the reuptake process. Now, in this particular slide, we're not using the word reuptake, are we? We're using endocytosis. Endocytosis is a synonym for reuptake or reabsorption. So, as I said, that's happening on the presynaptic membrane. What's happening on the postsynaptic membrane? Remember, the neurotransmitters, which are chemicals, are going to bind to receptors on the dendrites on the second neuron. Well, I said to you earlier that depending on whether the neurotransmitter is an excitatory neurotransmitter or an inhibitory neurotransmitter, the voltage of that second neuron is going to be altered. If we have an excitatory neurotransmitter, let's say we have glutamate, right? Glutamate is a primary excitatory neurotransmitter. Glutamate binding to the receptor, because it's a positively charged or excitatory neurotransmitter, it's going to depolarize the second neuron. So an excitatory postsynaptic potential makes that second neuron more likely to fire, right? As we get closer and closer to threshold, it's going to make that neuron more likely to fire. That makes sense, right? So if resting potential is negative 70 millivolts, depolarization makes it less negative. Well, an excitatory neurotransmitter is going to make it less negative, which means it's going to depolarize that cell and it's going to make it more likely to fire. Now, the opposite occurs with an inhibitory postsynaptic potential. Let's say we used a neurotransmitter that is inhibitory. So a neurotransmitter like GABA. GABA, being an inhibitory neurotransmitter, makes the next neuron less likely to fire, right? So with inhibitory postsynaptic potentials, the second neuron becomes hyperpolarized or more negative than rest, which means it's harder to have an action potential, right? Think about it. Go back to the bank account example I said. Imagine you had a bank account that's negative $70. Well, if there's another debit of $10, are you closer or further away from zeroing your balance? Now you're further away, aren't you? So an inhibitory neurotransmitter makes the cell more negative than rest, similar to an additional debit making your bank account more negative than prior. So in order to get to threshold in this scenario I gave you, now you have to travel 30 millivolts, right? It has to be a difference of 30 millivolts to get the threshold when at rest it was 20. So I said a lot, but I'm going to keep it simple. Excitatory postsynaptic potentials are what happens when you have a neurotransmitter that depolarizes the cell. Excitatory postsynaptic potentials make it more likely to have an action potential, whereas inhibitory postsynaptic potentials occur when you have an inhibitory neurotransmitter bind to the receptor site and you have hyperpolarization of the second cell, making it harder or less likely to fire. So I said a lot and I summarized there. Any questions about postsynaptic potentials? Now, before I go to agonists and antagonists, I'm going to go back to that neuron. Let's take a look at this motor neuron again, right? Everybody see it on the screen? Okay. I said these branches are dendrites. You have billions of neurons, right? It is estimated about 100 billion neurons in your brain. So you have all these neurons closely aligned. Do you think you have one neuron binding to the other? No. In fact, let's imagine we had neurons connecting to all of these branches. Some of those neurons are going to release excitatory neurotransmitters. Some of them are going to release inhibitory neurotransmitters. Now, what's the cell body got to do? It's got to calculate it all and say, okay, I received a little GABA, I received a little glutamate, I received a little dopamine, and I'm making it up, right? Okay, how much positive, how much negative do I have in total? And then it determines whether it hit threshold. So it's almost like, let's say you have Amazon. Well, Amazon's server is processing hundreds of thousands of charges at the same time. So are your dendrites. So they're sending a message, so is your soma. They're processing many messages from other neurons in the cell body. So let's go back to the concept of an agonist and an antagonist. These are also important concepts for you to understand. Now, what I like about this picture is neurotransmitters are three-dimensional structures. They're not two-dimensional, right? So you can see the three-dimensional nature of a neurotransmitter. Now, an agonist is a kind of molecule that mimics the process of a neurotransmitter. Now, in the pink, that is the agonist. In the brown, that is the receptor site. Now, in order for a molecule to bind to a receptor, it has to have a similar enough structure to the receptor site, right? So if you look at this agonist, it has two squares, no triangle. The receptor site was two squares, one triangle. Now, this is a cartoon. It's not exactly how it looks like in the brain. But because the agonist is similar enough, it's going to mimic the process of another chemical in the brain. So let's take morphine. Morphine mimics the process of endorphins. In fact, if you look at the word and break it into two, it's endorphin, endogenous morphine. Our body naturally produces painkillers. But how does morphine work? So if you take morphine as a function of surgery or whatever it might be, that chemical of morphine that you ingest is close enough to the receptor site to cause a release of endorphins, thus reducing pain, right? So think about that. That's an agonist. So an agonist mimics a neurotransmitter. Now let's talk about an antagonist. Now an antagonist, it blocks a neurotransmitter, right? So same receptor, different chemical, right? So if we were to look at the shape of the receptor, again, square, square, triangle, right? But in this case, the antagonist has one square. But even though it has one square, it still can link to that receptor because the shape is similar enough, right? And because it's similar enough, the neurotransmitter receives it and says, okay, I'll hold on to it. And similar to if you have a bathtub and you put the plug in the bottom, the water can't leave. An antagonist blocks the receptor site, making it impossible for other neurotransmitters to bind. And what that does is it interferes with other chemical messaging. Now, let's imagine this was an acetylcholine receptor. Now, acetylcholine is responsible for muscle flexion and extension in the body. But if we had a molecule that was similar enough that binds to the acetylcholine molecule, you can stop muscle movement. And in fact, that's what the neurotoxin curare does, right? So you might have heard poison darts. What it does is it freezes or blocks the acetylcholine receptors. Pretty cool. Botox is another example of that. But it operates as an antagonist. No other molecules, no acetylcholine molecules can bind to that receptor because the curare is there. Does that make sense? So it's pretty cool when you understand how the receptors work with the neurotransmitters. Now, again, so some of you are going to tell me, but hey, wait, I learned in biology it's also possible to have a partial agonist as well. Yes, again, these are the basics of chemical messaging. This is not to the same degree as you might learn in a pharmacology class. Okay, let me stop my share, and I want to show you a video which highlights how the neurons work and communicate. So let me open it up. Can everybody see the video? Yes?
Yeah.
Yeah.
So it's a brief video, but it explains a lot of the concepts I've said in short form and does a demo at the end. Here we go.
Our thoughts, memories, and emotions, as well as our sensations, perceptions, and actions, are facilitated by intricate networks of billions of neurons throughout the nervous system. These neurons communicate with one another via the transmission of electrical signals, or impulses. However, neurons don't actually touch one another directly. Instead, a tiny, fluid-filled synaptic gap separates the axon of the message-sending neuron from the dendrite of the receiving neuron. When a neuron transmits a signal, an impulse propagates along the neuron's axon. When the impulse reaches the axon terminal, chemicals called neurotransmitters are released into the synapse. Keep in mind that the dendrites of one neuron may receive signals from the axons of many other neurons. The right combination of neurotransmitters received by its dendrites will trigger a neuron to send an impulse of its own. In this animation, an impulse is represented by a green ball of light. The impulse travels along the axon until it reaches the terminal. There, it causes neurotransmitters to be released into the synapse. In this animation, neurotransmitters look like red balls of light. Neurotransmitters allow an impulse to cross from the axon of one neuron, across the synapse, and onto a receiving dendrite on another neuron.
Now, what I showed you that video is because when I'm describing with words, it may not, it may not make as much sense. But as you saw, those beams of light moving down the axon and then ultimately connecting with the dendrites on the other side, that's how the chemical messaging process works. That's how you have communication between neurons. And one of the things that I loved about this particular video is it reminds you that another neuron is receiving impulses from many other cells. So it has to process all of that information before determining whether it wants to have an action potential of its own. And the soma has to figure all that out. But if after all of that input hits threshold, you will have an action potential. So let's stop there and we'll pick up after the break.